Ribosomes are the protein synthesis units of a cell. Ribosomes are small, dense, rounded, and granular particles of the ribonucleoprotein. They occur either freely in the matrix of mitochondria, chloroplast, and cytoplasm or remain attached to the membranes of the endoplasmic reticulum. They occur in most prokaryotic and eukaryotic cells and provide a scaffold for the ordered interaction of all the molecules involved in protein synthesis. They are the most abundant RNA protein complex in the cell, which directs the elongation of a polypeptide at a rate of 3 to 5 amino acids added per second. The ribosomes occur in both prokaryotic and eukaryotic cells. In prokaryotic cells, the ribosomes often occur freely in the cytoplasm or sometimes as polyribosomes. In eukaryotic cells, the ribosomes either occur freely in the cytoplasm or remain attached to the outer surface of the membrane of the endoplasmic reticulum. The yeast cells, reticulocytes or lymphocytes, meristematic plant tissues, embryonic nerve cells, and cancerous cells contain a large number of ribosomes which often occur freely in the cytoplasmic matrix. Cells like the erythroblasts, developing muscle cells, skin, and hair which synthesize specific proteins for intracellular utilization and storage containers also contain a large number of free ribosomes. In cells with active protein synthesis, the ribosomes remain attached to the membranes of the endoplasmic reticulum. Structure. The ribosomes are oblate spheroid structures of 150 to 250 A ring in diameter. Each ribosome is porous, hydrated, and composed of 2 subunits. One ribosomal subunit is large in size and has a dome-like shape, while the other ribosomal subunit is smaller in size, occurring above the larger subunit and forming a cap-like structure. The small ribosomal subunit contains a single RNA molecule, referred to as small RNA. The large subunit contains a molecule of large RNA and one molecule of 5S RNA, plus an additional molecule of 5.8S RNA in vertebrates. The lengths of the RNA molecules, the number of proteins in each subunit, and consequently the sizes of the subunits differ in bacterial and eukaryotic cells. The assembled ribosome is the 70S in bacteria and 80S in vertebrates. There are great structural and functional similarities between ribosomes from all species, which is another reflection of the common evolutionary origin of the most basic constituents of living cells. The 70S ribosome consists of 2 subunits, 50S and 30S. The 50S ribosomal subunit is larger in size and has a size of 160 A ring to 180 A ring. The 30S ribosomal subunit is smaller in size and occurs above the 50S subunit like a cap. The 80S ribosome also consists of 2 subunits, 60S and 40S. The 60S ribosomal subunit is dome-shaped and larger in size. In the ribosomes which remain attached to the membranes of the endoplasmic reticulum and nucleus, the 60S subunit remains attached to the membranes. The 40S ribosomal subunit is smaller in size and occurs above the 60S subunit, forming a cap-like structure. Both the subunits remain separated by a narrow cleft. The 2 ribosomal subunits remain united with each other due to the high concentration of the Mg++.001M ions. When the concentration of Mg++ ions reduces in the matrix, both ribosomal subunits get separated. Further, during protein synthesis, many ribosomes are aggregated due to common messenger RNA and form polyribosomes or polysomes.